So I just wanted to start off. This is one of my favorite slides. Can everybody see my screen? Okay. I start off every cure I teach with this slide because it really encapsulates the process that's going to happen during the course of the semester. Some famous educators said many years ago, many high-impact educational practices tend to involve learning at the edge of chaos. And I explained to the students that my job is to make the chaos manageable, not to prevent the chaos. And we go from there. So the group of potential projects that I'm going to talk about for the next, I've got 3 hours, Joe? 10 minutes, all involve non-covalent interactions, and the theme is non-covalent interactions. And one of the things I always do early in the class to set up that connection with the basic chemistry, biochemistry, is we talk about the role of non-covalent interactions in enzyme structure and function. In general, the sorts of things that involve non-covalent interactions. And we go through this mind map. I'm not going to spend any time doing it because you all know all the different things that non-covalent interactions are involved with. But I think it's important to set a tone that, we're really thinking about fundamental chemical properties early in the course. So the title I gave Joe, I think, for this was Understanding the Roles of Non-Covalent Interactions in Structure-Function Relationships in Malate Dehydrogenases. And these are projects involving the loop region and or subunit interfaces and subunit region, interface regions, focusing on roles that non-covalent interactions play in malate dehydrogenase. Now, on the website, and this is password protected. If you click on this, you'll find that it asks you for a password. You can get the password from me very easily just by emailing me. The intent is to provide useful information for faculty that are thinking of incorporating these, not to provide one-stop shopping for students that have got to write a paper or write a proposal or whatever. So the goals of this set of cures, and I'm going to talk about 6 different areas and ideas to be incorporated into cures, is to provide, a cure experience that incorporates all 9 aspects of research with a goal of enhancing both technical and soft skills that both academia and the biotech industry value. And I think sometimes we overlook the fact that these guys, graduate schools, biotech industry, jobs, these are the end users of the students that we're producing. And then, of course, as we've all emphasized, is to generate publishable science of broad interest. So if you're interested in any of the 6 project areas that I'm going to talk about in the next few minutes, and would like to be involved, and you'll see some of them have got scientific papers in progress, some of them have got plans for new papers, etc. Contact me, we'll set up a Zoom. There's lots and lots of collaboration opportunities out there for this. Okay, so these are the 6 areas. So Jess and I talked about this a lot, and we divide them into 6 areas. The role of the mobile loop in ligand specificity, understanding the catalytic mechanism of malate dehydrogenase, subunit interfaces and allostery, oligomeric structure and stability, understanding the isoform differences and biological roles of malate dehydrogenases, and then this whole area of drug design where the cures are based upon pathogens and hosts and thinking about how to design orthosteric or allosteric inhibitors of these proteins. So all of these areas can involve as many of these types of experimental approaches. Most of them involve some sort of mutagenesis, expression, and protein purification. Though there are a lot of mutants available now that you can skip the step of making, and sort of, if you want to, it just depends upon your course. So, you can look at this and go, gee, I really want to get enzyme kinetics into it, or I really want to get, and all of these cure areas are amenable to these. And there's some nice collaborations, the instrument collaboration type things that we talked about yesterday, and you'll hear more about that because Chris is going to talk about some of the collaborations that he's set up later in the day. So the first area, the role of the mobile loop in ligand specificity. So everybody talks about the mobile loop, but here is a picture, and on the website there's a Pymol PSE file that you can download and look at and play with. But the open loop, you have the open loop before anything bound NAD comes in, and it partially closes the loop. Malate comes in and it closes it a bit further, the green line. So the loop changes. You'll notice this helix up here doesn't change as the loop closes in the active complex, but you put in citrate, which is where the red loop is, the loop changes even more, and you can think about what's involved in those loop movements, and think about what are the controlling features of the loops and things like that. We've done a lot of work on this. The loop is involved in ligand specificity. There's the substrates, there's alternative substrates, there's oncometabolites that are interesting to binding in. So this whole idea of substrate specificity and inhibitor specificity. We've actually got several, one fairly advanced manuscript in preparation. Some of you are involved in that because you've contributed information about loop mutants and things like that you've either created or you've used from the library of loop mutants. And there's a series of abstracts that have been published over the last few years about some of these things that are listed here. And again, these are all things that are easily findable if you do the right sort of literature searching. But for faculty, we put them here to get around that. Okay. The second area is thinking about the catalytic mechanism. So this derives from the idea of the catalytic mechanism was first proposed by Len Banaszak, who in many ways is the godfather of MCC, as we talked about yesterday. But there's a histidine in the active site. My mouse is not behaving. There's a histidine in the active site here. There's an aspartic acid that changes the basicity of the histidine. The histidine abstracts a hydrogen from the malate or donates a proton to oxaloacetate, depending on which direction the reaction is. There are two arginines that are known to be involved in binding. There's a third arginine in the active site that's completely conserved that doesn't seem to be directly involved in binding, but may be involved in helping to steer the substrate in and out of the active site, things like that. Lots of questions we do not know the answers of. And so, for instance, if you want to think about things with this and you want to overlap with Joe's exciting stuff about metabolon formation, how things get in and out of the active site of malate dehydrogenase and into the active site of citrate synthase are really interesting questions that Joe's working on. And so, there's overlap between this sort of aspects and what Joe's trying to do with metabolons. Again, there's a group manuscript in process with this. And we've been interested and had lots of abstracts over many years now, dating way back to the 1990s, about the role of what are called second sphere residues in catalysis and how those alter what's going on in the active site. So, these are what we can call the first sphere residues, but there are a whole slew of residues around that are thought to modulate what's going on in the active site. So there's lots of interesting potential projects thinking about the catalytic mechanism. The third area is subunit interfaces and allostery. This is an analysis of the dimer interface in malate dehydrogenase using a computational approach called HINT. It was actually first developed for looking at hemoglobin subunit interactions, but it literally maps every atom-to-atom contact and tells you what things interact with one another, not necessarily by touching, but through electrostatic interactions or whatever across the interface. We've got all that HINT data for several different MDHs, different complexes, and are happy to share that data with people that are interested in exploring the interface. But again, there's multiple roles. If you've followed what we've been doing in the last 10, 15 years, you'll know we've looked at a number of interactions, number of residues across this interface. And again, we're in the final stages of preparing a manuscript. The emphasis is twofold. One is the multiple roles of the interfaces. And I've talked about the dimer here. We'll talk about the tetra interface as well a little bit later on. But what we're really interested in is what I call triggers and transmission, i.e. how does something happening on one active site transmit information to a different active site, and what's involved in that. So again, lots of cool projects that can be developed and get involved with those.
2-minute warning.
Minute. Oh, I better speed up. Okay, so we're interested in the tetramer and dimer questions. Again, lots and lots of cool projects. I'm going to speed up here because I really want to spend my minute, if I've got it, talking about this one. This is one I'm particularly excited about. And this is one you'll see there's no abstracts, no manuscripts in preparation. These are projects in preparation, but looking at understanding isoform differences and their biological roles. And the Arabidopsis, the watermelon, and tobacco, which we're developing a bunch of expression systems for the tobacco systems at the moment in conjunction with some people in the biology department. There are cytosolic, mitochondrial, chloroplastic dioxysomal forms. All these, some of them have different cofactor specificities. There are projects to think about tissue localization, the differences between precursor and mature isoforms. And those of you that have looked at our first paper on the structure of the watermelon know that it's a comparison of the precursor form and the mature form and the enzymological differences between those. So there's lots of cool projects that can be developed with developmental biology, making transgenics, looking at systems biology, looking at metabolomics. We picked Arabidopsis and tobacco just because they're both easy to work with. It's easy to make transient transgenics of tobacco leaves and look at things. These are a little bit longer, but again, there's well-known technology and they're easy to make transgenics. So that's a sort of a whole new area that we're branching into, to marry the basic enzymology differences with a more biological perspective. And then the final one is, we've talked about this a lot. We've talked about the malaria one, drug design, so I won't spend much time on this. Again, there's a manuscript in preparation. We've got 3 different pathogenic species we're looking at in the cures that I'm teaching, and some that we're collaborating with some other folks on. Plasmodium, Candida albicans, and Porphyromonas gingivalis. These 2 came out of student-generated interest in a cure a couple of years ago, where the student says, gee, I want to be a dentist. Why can't we work on something that's related to dentistry? And so I challenged them to think about the oral microbiome and things they might be able to do with that. And they came up, and we're now, developing a cure project around those. So I'm sorry if I went